Professor Ohad Gal-Mor always knew he wanted to be a scientist. His fascination with microbiology grew as he became more cognizant of the sophistication and complexity of bacteria. His breakthrough research on Salmonella contributed greatly to the worldwide scientific community regarding virulence mechanisms and regulations of pathogenicity and their role in Salmonella and other pathogens infections. The Talpiot Medical Leadership Program is proud to partner with the Nehemiah Rubin Excellence in Biomedical Research, the TELEM program at Sheba, which is under the directorship of Professor Gal-Mor, and we are delighted to host him. Professor Gal-Mor still has his passion for the great outdoors and enjoys hiking, biking, and swimming. Talpiot XMed, Leadership in Healthcare.
Good evening. Today I want to talk about the phases of the antibiotic-resistant global crisis and possible ways to fight it. Bacterial infection resistant to antibiotics is one of the most serious and severe challenges that the modern medicine is now facing. On a global scale, each year, 700,000 people die from resistant infection, and estimations suggest that by 2050, this number will increase to 10 million with an annual economic burden of $88 trillion US. This dramatic and global crisis is rapidly escalating because of three main reasons. The first reason is very low investment by the large pharmaceutical companies in the research and development of new antibiotics due to low profitability. The second reason is overuse of antibiotics in the agriculture sector, when these are often being used as prophylaxis and animal growth promoters. And the third reason is extensive misuse of antibiotics in humans. About 2/3 of all antibiotics treatment in the USA are incorrectly prescribed. But how antibiotic resistance is actually acquired? In bacteria, antibiotic resistance can be acquired by different mechanisms. However, the most efficient and alarming way is by a process known as conjugation. Please remember this term, which simply means sex between bacteria. In this process, a mobile genetic element known as a plasmid is being replicated and then transferred from a donor bacterium to a recipient bacterium via a special tube called conjugation bridge. This is how the conjugative bridges look like under a powerful electron microscope. These tubes are used for the transmission of resistance and virulence genes carried on mobile genetic elements between bacteria. Therefore, this process plays a key role in the evolution and emergence of new bacteria populations that can be not only multi-drug resistant, but also hyper-virulent. This sounds very scary, and it is indeed a powerful driving force in bacterial evolution. But what if I tell you that we can control and restrict this process? Work done in my lab in the last few years has shown that bacterial conjugation could be limited by certain chemical compounds, specific physiological conditions, and surprisingly, even by microgravity. In the lab, we can mimic, to some extent, microgravity using a special device that was developed at the NASA's Johnson Space Center to simulate the effects of microgravity on cells. Experiments with this device suggested that under microgravity-like condition, bacterial conjugation significantly decreases by about 40%. And from here, the only way was up. To further test these effects of microgravity, we have recently used the lab-on-chip platform developed by Space Pharma and the nanosatellite Dido-3 to send bacteria to space and study how the space environments affect their conjugation. The original launching time was scheduled to October 2019. However, a mission that should have been launched before ours, using the same Vega rocket, crashed and everything stopped. After the rocket crash, the COVID-19 pandemic has started to spread in the world, and they had to shut down the launching facility at French Guinea. After that, we faced a long period of bad weather with windstorms at the high level of the atmosphere, and the launching was cancelled and suspended over and over again. After 10 months of nerve-breaking anticipation, when we started to lose hope, the satellite was finally launched. This happened on September 3rd at 5:35 a.m. Israeli time, carrying our patiently waiting bacteria to space.
3, 2, 1, top! Allumage P80. Allumage P80, décollage.
Our experiment in space at an orbit of 500 kilometers above Earth lasted more than 40 days. During this exciting time, we were able to closely follow bacterial conjugation and the rate of transferring resistance genes in space. Comparison of this space experiment to an identical ground experiment that was conducted in parallel indicated that conjugation in space was decreased by about 2,000-fold in comparison to on-Earth condition. Okay, so this is all very nice and neat, but one may ask, what do we care about bacterial sex in space? Well, we do care about it because of two main reasons. The first one is because when we send people to space, we actually ship more bacteria than human. The number of bacteria in our body exceeds by more than threefold the number of all human cells. And we want to know how microgravity and space environment may change the resistance and virulence potential of our own microbiome in space. Currently, we send on a regular basis astronauts to the International Space Station. But even now, given that you have enough money, you can purchase a space traveling ticket for recreational purposes from different companies like Blue Origin, Virgin Galactic, or SpaceX. Of course, this trend of space tourism will become more popular and more frequent in the not that far future. The second reason this is interesting for us is that it may teach us how to fight conjugation also on Earth. The influence of microgravity on conjugation is most likely due to the effect of microgravity on the assembly of the conjugative bridge I showed you before. Therefore, we reasoned that if we can find other ways to interfere with the assembly of these conjugation bridges, we could risk conjugation and the dissemination of resistance virulent genes between bacteria. In other words, we can possibly use this gained knowledge to develop contraceptives for bacteria. One material we found to do exactly that is a compound which is naturally present in bile and called glycoholic acid. We found that glycoholic acid significantly inhibits conjugation and limits transfer of resistance and virulence genes between bacteria, as you can see here. Therefore, we propose to use these or similar compounds in hotspots like hospitals or agricultural facilities as an innovative way to fight the fast dissemination of antibiotic-resistant genes, which results in hypervirulent and hyperresistant new bacteria strains. When I have opened my lab at the Sheba Medical Center, I have never imagined, even in my wildest dreams, that we will send bacteria to space. But the last unusual year has proven that for us, even the sky is not the limit. Thank you very much.
Talpiot XMed, Leadership in Healthcare.